Hello, Beth.
Hello, Erin. Always lovely to see you.
I agree. Yeah, so recently we actually gave a quick overview, well, as quick as I'm able to do when
As
I'm,
we, yeah.
of the cell painting assay. And so today I thought it would be interesting to dive into like what do cell painting features mean? There's a lot of discussion in particularly, right now in morphological profiling, which again, as I sort of said in the cell painting episode, is kind of like the omics of looking at cell shape that we're doing in cell painting assay.
Yeah.
And
Shape and intensity and textures and crazy things and
yes,
yeah.
yes, absolutely. Yeah. But in our, I mean, I think in probably in every field, but I can only speak knowledgeably about our particular field. There's lots of discussion at the moment of like, okay, everybody wants to do deep learning. And one of the major drawbacks of many deep learning techniques is that the features that come out at the end that are, describing your condition are not interpretable. And so the cell painting assay and the sort of more traditional features that we're getting out of CellProfiler are quite interpretable. Um, and so I thought it would be helpful, though, to kind of like dive into the feature names
Oh
and
yeah.
help explain, like, great, people say, oh yeah, CellProfiler is interpretable, but like, how do we actually interpret them? Let's let's look at them. So, yeah. With that, I am sharing here. This is a page in so the 2023 Simony Nature Protocols paper was a giant work led by my dear Beth right here.
Mhm.
That was optimizing the cell painting assay. So this is the second time that we had a major protocol paper published on the cell painting assay. But here in this repository, and we'll drop the link in show notes, we have a wiki that has just sort of some extra, like casual, more casual information about the cell painting assay. And one of the things we have in here is what do cell painting features even mean? And so I thought this was just a great thing to like scroll through, chat through, and
Mhm.
explain what they mean.
Yeah.
Um,
Well, and because I'm incapable of taking credit for anything, this is basically copied verbatim from like the wiki for the cell painting V2 paper and was built over like many years by lots of people. This is not a thing that I personally made, but it is a really great resource that the lab has made over the years.
yes, yeah. Um, all right, fine. You can refuse to take sole credit on this. Didn't mean to imply, yeah, did not mean to imply this was all
Hmm.
Beth's work because so many
Yeah.
fabulous, wonderful scientists have worked
Mhm.
to lead us to this point.
Yes.
But it is in a repository
Mhm.
named for the paper that Beth led. So
That is true.
I'm just putting that out there. Okay, here we go. So if you need a refresher on the cell painting assay, go check out the that episode. We'll drop it in show notes. Um, but basically, as a reminder, we have pictures of cells, we identify nuclei, we identify cell bodies, we define the cytoplasm, which is the cell minus the nucleus, and then there are a number of channels that we are making measurements in. So we are identifying the cell, for example, and then making a whole series of measurements using that same object. So one thing I want to emphasize here is that the object that we are identifying, we may use some combination of channels for identifying like a cell object, but then that object is independent of the channels that we used to identify it. It is just now like a mathematical squiggle
Mhm.
right? And we're saying, okay, now I can place that squiggle on top of any of these images and make measurements about what is inside that squiggle.
Mhm.
Sorry, in any of these images independently of what
Mhm.
I used to create that cell object.
Yeah, that's a super important point to emphasize.
Yeah. So we have these 5 channels. We give them names. Usually we name them based on what we are looking at, the expected structure. So DNA, RNA, ER, mito, AGP, standing for actin, Golgi, and plasma membrane. So that is our description for the image itself, because it's the structure that we are expecting to see in that image based on the stain that should be showing in that channel that we imaged. Um, we have right. So then many measurements, thousands of measurements.
Mhm.
And
Yeah. This is an older version because back then we were only doing like 2,000 features, and
yeah,
now we tend to do closer to like 8,000.
yeah, yeah. It's kind of nuts. All the things. Um,
I like it.
yeah. So, um, we then again we take measurements in individual channels in the different objects, and then we can also make extra measurements, right, that are features looking at the relationship between channels. So that's an extra set where we then have correlation features that then are describing multiple channels, the mathematical relationship between information in multiple images within those objects. So, yeah, I mentioned we have the cells, the cytoplasm, and the nuclei as our objects, and then we have a bunch of categories of features that we're measuring. So these are links to the CellProfiler documentation telling you more details about the exact measurements in each of these categories. But just to note, there are different categories of measurements, and they can be things such as shape, area shape, describing that's not actually looking at fluorescent, and pixel intensities. It's telling you just about the shape itself. But then something like intensity, like that's telling us all about pixel intensities within that shape, and often um, includes things that are quite simple, like the total intensity or the mean pixel intensity within there. But it can also be more complex, like talking about the distribution or the patterns of those intensities. So sort of a full range of like very easily understandable to like actually I can wave my hands and tell you what it's doing, but it's
Mhm.
but digging into the very specifics is complicated. And then some of these like texture and granularity again are quite easily on a sort of conceptual level of we're looking at how much texture is in an image, and granularity is if I erode part of the image, a piece at a time, how much is left. So it's also kind of like a very similar to a texture conceptually, but mathematically quite different. And mathematically, you know, so we get the sort of like the biologist hand waving, oh, it's looking at texture, but in terms of like what does that mean? Uh, there can
Mhm.
be a bit of a disconnect. Like likewise, so I think also talking about interpretability, like these all are interpretable in that we can tell you the exact math. So lots of deep learning is a black box of what math is happening. Here, we can tell you the exact math. Like you can go to the cell profiler code, you can look and see like what are the numbers.
Yeah.
I mean, if you really want to,
Yeah.
what numbers go in and what numbers come out. Um, and but a biologist is going to have a much easier time telling you, like, oh, cell area. Like, I know exactly what that means versus, like, oh, the granularity, uh, you know, of this structure in this, like it gets a little more hand wavy what that could actually mean. So, but so that is, we have these feature categories, different kinds of measurements. Also, some of them have extra things tacked on the end. So like Zernikeys, those are a math a math for for
I was
describing.
gonna say maybe we should do a Zernike episode someday, but no, we probably should not ever do a
Maybe
Zernike
you could do
episode.
a Zernike episode. I mean, I do think like I think that would actually be awesome. It's really interesting what it's doing. I understand it to the level of it's telling us, how things are it is math that tells us like how things are arranged. Um,
I think that's all you need to know. I think you just did the Zernike episode.
no!
Congratulations.
But so, some features, right, have different parameters at the very end of the feature name saying like, okay, this is additional pieces of information you need to know mathematically, to understand the math. So like the scale at which these things are measured. Um, so that is like how we get to these features in a nutshell. And then um I just wanted to show, like I can show you really quickly um just a few examples, of we can just dive into a couple features here. Um doo, doo, doo. So, here is opening in Excel, so it's not the most beautiful, but if we, okay, let's see. I no, there we go. To make this readable, just picking some random features here. Um, Again, this is just totally at random, but the way that so all of the CellProfiler features are named in a consistent way. So it starts by telling you what compartment um, this was, what object this was measured in. So we measured it in the object we called cells. Then this is the category of measurement that we made. So we made a texture measurement. This is the specific measurement that we made. I don't know what an info measurement one is necessarily, other than it is one math to describe texture. um, then it's telling us the name of the image that we measured it in. So again, we named this image actin because it was the structure that we expected to see given our stains. You could name it you can name it whatever. There's many ways that one could name, but this is our standard for the cell painting. And then this is just some extra parameters that have to do with the info measure one. Oh, this is a fun example, and this leads right into what I wanted to say. Okay, so this is saying within the nuclei, we measured an intensity feature. That specific feature is the standard deviation of the intensity at the edge of the object, and we measured it in the actin channel. But wait, you might say, there shouldn't be any actin within the nucleus. What? That doesn't make any sense. And so here, I think this is a really important point that I want to make about cell profiler features. So we will get questions about this, like, okay, but how is that interpretable? You can't interpret actin in the nucleus. There is no actin within the nucleus. But wait,
Well, a little bit, but it shouldn't
okay,
be F actin. Shouldn't be
wait, it
phalloid and
shouldn't be F
actin.
actin. We should
Yeah.
not see F actin within the nucleus. So what I want to point out is here, this beautiful illustration I clearly spent hours on, but here,
I love it.
yeah, absolutely, not to scale. But if we think of, we have an objective and we have a cell growing on a cover slip. And so cells, even cells that are beautiful for microscopy that like pancake out nice and flat for imaging, even those kinds of cells, they are in 3 dimensions. So yeah, like,
We
whoa,
don't live in flatland.
cells are 3 dimensional. So what that means is, in this example, I have blue nucleus and I have some cellular compartment. We can pretend it's ER. I don't know, that's like perinuclear here.
I mean, it's red, so it's the mitochondria, but
I
sure.
know, I know.
Yeah.
Okay, right. I know.
Yeah.
Mito's red.
Yeah.
So, it's some unknown cellular structure here.
Yeah.
But if we look at that from the top, what we see is that um, so we have, can clearly see the nucleus is this nice round thing here in the middle of the cell. From the top, the majority of the signal, yes, the majority of this red is going to show up as in a circle around the nucleus. But because this cell is 3 dimensional, there is actually some structure that when we are, looking sort of this
Mhm.
top view, or I guess a bottom view, because we have the objective
Yeah.
bottom here, bottom view of the cell, um, you do see some
Mhm.
of this
Mhm.
compartment that is outside of the cell,
Mhm.
looks like it's overlapping with the nucleus. And so when we're looking at features here, these measured features,
Mhm.
so Hoechst tends to be a very nice, tight stain that doesn't drift cytoplasmically. So we'll see, the intensity of our DNA within the cell, we basically see nothing. And
Mhm.
if we look at the intensity of the DNA within the nucleus, it's very, very bright. And
Mhm.
if we look here at my pretend ER, that even though it's ER should be, green and mito or red,
Yeah.
bear with me. So
Erin and I have very particular feelings about what color different
I
organelles
know
are.
it's kind of ridiculous, but also like, yeah, it is what it is.
Yeah.
So then if we look in the cell, we see, right, like clearly that makes sense. We see the ER here is mostly perinuclear, like that's biologically true. But then if I look at features measured in the nucleus, right, because it is this object that is a circle of a three-dimensional compressed thing, like we're seeing
Mhm.
some of this stain is actually
Mhm.
there in our nucleus object, which doesn't reflect completely real biology,
Mhm.
nucleus, like in
Mhm.
the cell. We don't have endoplasmic reticulum inside of the nucleus in a cell, but our stain, because we're going from 3D to 2D measurements,
Mhm.
we will measure some there. So that's how to interpret if you get this sort of nonsensical feature. Um,
Mhm.
well, but yeah, go, Beth.
Can you go back to I just want to make one microscopy point,
Yeah.
which is like, and some of this has to do with sort of how thick of what's called an optical section that we're taking.
Yes. Yeah.
So we tend to do cell painting in wide field, though you can do it in either wide field or confocal. And what that means is if I draw on Erin's lovely drawing here with my annotator, um, with wide field microscopy, you don't tend to get a lot of Z resolution. You tend to take light from a lot of different Z points in your sample. And so that's why we have this effect. If we really, 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 really only wanted a very thin one, if we did something like confocal microscopy, we could take a thinner Z section and then we wouldn't see much or at all of this stuff that's on top of the nucleus. But for cell painting, we typically want to gather as much information as possible. And so that's why generally we prefer to use wide field so that we get more of the stuff, even if it makes it a little bit more jumbled because we're seeing a 3D thing compressed into 2D. But it is totally possible to take a thin section where this doesn't happen if that's what you want for whatever assay you're doing.
Yeah, absolutely. Good point. And we can go like we'll also coming up soon have an episode about like how can you modify the cell painting assay and and talk about wide field versus confocal. But in terms of, what is our standard cell painting assay is wide field. So we are getting all of this extra information. But like again, great point. It could be handled differently, such that you could end up with a different way of imaging here. This is just not what we're usually working with. Um, yeah, it's a great point. And I think
Yeah.
that's what I wanted to say about cell profiler features and understanding and interpreting them.
Yeah. No, and definitely some of them will give you a picture in your head. Like if I say my cell got has an area 2 times as big, to your point earlier, like some of the things definitely like the picture, like the info measure, it's like, okay, the texture is kind of something about this, but I don't really, but it should be enough to at least give you an idea of like what's going on in the cell to try to give you an idea of what's the next experiment to do.
Yes, yeah, and certainly way more informative than feature 732, which might be the name of a feature you would get out of a deep learning model, right? Like,
Yeah.
so there is definitely always some level of interpretability because
Mhm.
what image, what compartment, and
Yeah.
what category of measurement
Mhm.
it is, even if the
Yeah.
math to get there is
Mhm.
a
Yeah.
bit maybe.
Awesome. Well, thank you. That was great. And I love your illustration.
Thank you. Thank you. Thanks for being here. Thanks for joining. We'll talk to you
See you
later.
later.